Every single one of us is walking around with genetic material that neither of our parents actually gave us. Dozens of tiny changes buried inside our DNA that appeared out of nowhere. And for decades, scientists could not fully explain where they came from or why some families produce far more of them than others. This is not science fiction, and it is not a hidden government experiment. It is one of the strangest and most quietly unsettling facts in modern genetics. The discovery that every child is born carrying a small collection of brand new genetic variants that trace back to neither their mother nor their father, changes that essentially appeared out of thin air somewhere between conception and birth. The number is not exactly 348, and it is not some unsolvable mystery either. But what researchers have actually found through years of genome sequencing is arguably even stranger than the viral headlines suggest, because it means every human being is, in a very real sense, a walking genetic experiment that has never happened before in exactly the same way. Scientists call these unexplained genetic changes de novo mutations, a term that literally means from new, and they represent one of the most fascinating quirks of human reproduction. When a child is conceived, half of their DNA comes from their mother's egg, and half comes from their father's sperm. And in theory, every single piece of genetic code in that child should be traceable back to one parent or the other. In practice, that is not quite what happens. Large-scale genome sequencing studies, some involving dozens of families where scientists sequence the complete genetic code of both parents and their children side by side, revealed that children are consistently born with somewhere between 40 and roughly 100 brand new genetic variants that cannot be found anywhere in either parent's DNA. These are not typos in the sequencing machine, and they are not contamination errors. They are real, physical changes to the genetic code that were not present in either parent, yet somehow ended up in their child. A process that involves copying enormous amounts of genetic information with almost unbelievable precision. The human genome contains roughly 3 billion individual chemical letters, and every time a cell divides, all 3 billion of those letters need to be copied accurately so the new cell has a complete and correct set of instructions. The molecular machinery responsible for this copying is remarkably good at its job, but it is not perfect. And across billions of letters copied over and over throughout a person's lifetime, occasional small errors slip through. Most of these copying errors get caught and repaired by other specialized molecules whose entire job is to scan freshly copied DNA and fix mistakes before they become permanent. But every so often, a mistake survives that repair process, gets locked into place, and if that mistake happens to occur in the specific cells that eventually become sperm or eggs, it can end up being passed directly into the next generation as a brand new mutation nobody else in the family has ever carried. What makes this story genuinely interesting rather than just a technical footnote is the discovery of exactly where most of these new mutations come from, and the answer surprised even the scientists studying it. Research comparing full genome sequences across large numbers of family trios, meaning a mother, a father, and their child, all sequenced together, found that the overwhelming majority of these unexplained new mutations originate from the father's side, not the mother's. And the reason comes down to a basic biological difference between how men and women produce reproductive cells. A woman's eggs are essentially all formed before she is even born, meaning the genetic material inside them has been sitting relatively dormant for decades by the time she conceives a child. A man's body, on the other hand, continuously produces new sperm throughout his entire adult life, which means the cells that eventually become sperm keep dividing and copying their DNA over and over, again for years. And every single one of those copying events carries a small risk of introducing a new, uncorrected error. This single fact leads directly into one of the most well-documented and genuinely consequential findings in this entire field, the relationship between a father's age and the number of new mutations he passes on to his children. Large-scale studies sequencing the genomes of dozens of Icelandic families found a remarkably consistent pattern. Every additional year of a father's age at the time of conception adds roughly 2 additional new mutations to his child's genome, meaning older fathers pass on measurably more of these unexplained genetic changes than younger fathers do. Researchers calculated that the number of these paternal mutations effectively doubles for roughly every 16 years of paternal age, which means a child, born to a father in his 40s, may carry noticeably more of these unexplained genetic variants than a child born to the same father in his 20s. Maternal age plays a much smaller but still measurable role, with mothers contributing a modest additional handful of mutations as they get older as well, though nowhere near the scale seen on the paternal side. None of this would have been discoverable at all without the arrival of whole genome sequencing technology, and this is really where the story becomes a story about modern science catching up to a biological reality that had been invisible for most of human history. Before researchers could sequence entire genomes quickly and affordably, there was simply no practical way. to compare a child's complete genetic code against both parents at the level of detail required to spot these subtle new variants. It took large, carefully designed studies, sequencing entire family trios, and comparing every single letter of genetic code across 3 complete genomes at once to finally reveal just how common these unexplained mutations actually are. One especially detailed study involving a 4-generation family with dozens of members mapped out these new mutations with extraordinary precision, finding that each new generation picks up somewhere between roughly 100 and 200 new genetic changes when every type of mutation, not just simple letter swaps, but small insertions, deletions, and repeated sequence changes, is counted together. It would be easy to assume that carrying dozens of mutations nobody else in your family has ever had sounds alarming, but the reality is far less dramatic than it sounds. And this is the part of the story that tends to get lost in more sensational retellings online. The vast majority of these new mutations land in parts of the genome that do not code for anything important, essentially harmless. Genetic noise that has no measurable effect on a person's health, appearance, or abilities. Only a small handful of these new mutations, often just 1 or 2 per person, land inside an actual functioning gene where they could potentially have any effect at all, and even then most of those effects are neutral rather than harmful. This is actually one of the most important and reassuring findings to come out of decades of studying de novo mutations, that the human body has evolved multiple layers of protection specifically because a small number of unpredictable new mutations every generation is simply an unavoidable cost of how reproduction works, and the system is built to absorb that cost without most people ever noticing any consequence at all. That said, there's a smaller but medically significant fraction of cases where these unexplained new mutations do matter enormously, and this is where the science becomes genuinely important rather than just a curiosity. Researchers studying conditions like autism spectrum disorder, schizophrenia, certain congenital heart defects, and a range of early-onset developmental disorders have found that de novo mutations play a measurable role in a meaningful percentage of cases, particularly in children who develop these conditions, despite having no family history of them whatsoever. This finding actually solved a long-standing puzzle in medical genetics, because doctors had struggled for years to explain why conditions like autism and schizophrenia continue to appear at a fairly constant rate across generations, despite carrying real reproductive disadvantages that, in theory, should cause harmful genetic variants to gradually get filtered out of the population over time. The discovery of de novo mutations provided a compelling answer. Since these conditions do not need to be inherited from a parent who already carries a harmful variant, they can arise, fresh in a single child, through a brand new mutation that appeared for the very first time in that specific pregnancy, which means the disorder never actually needs to be inherited at all in order to keep appearing generation after generation. The connection to paternal age adds another layer to this picture that has real implications for modern families, especially as more people around the world are choosing to have children later in life than previous generations did. Multiple large-scale studies have found a genuine statistical association between advanced paternal age and a modestly increased risk of conditions like autism and schizophrenia in offspring. And for years, the leading explanation was that this increased risk came directly from the higher number of de novo mutations older fathers pass on. More recent and more detailed research has actually complicated this picture in an interesting way, suggesting that while de novo mutations do contribute to this risk, they may only account for a relatively small portion of the total increased risk associated with older fatherhood, meaning other factors, possibly including subtle chemical changes to how genes are switched on and off, rather than changes to the genetic code itself. Likely play an important role as well. And this is a great example of how real science tends to work. An initial finding generates a clean and satisfying explanation, and then years of follow-up research reveal the true picture is more complicated and more interesting than the first headline suggested. Beyond disease risk, there is also a genuinely fascinating evolutionary angle to this entire phenomenon that rarely gets mentioned in the more sensationalized versions of this story. These unexplained new mutations are not just a quirky biological accident, they are actually the fundamental raw material of evolution itself. Every single genetic variation that has ever existed in any species, every trait that has ever helped an organism survive and reproduce more successfully than its neighbors, originally arose as exactly this kind of unexplained new mutation in some ancestor, appearing for the very first time with no family history behind it whatsoever. Without a small, steady trickle of brand new genetic changes appearing in every generation, evolution as a process would grind to a complete halt, because natural selection can only work with genetic variation that already exists, and de novo mutations are the primary source of genuinely new variation entering any population. In other words, the very same biological process responsible for a small percentage of developmental disorders is also the exact same process. responsible for the entire diversity of life on Earth, including every adaptation that has ever allowed any species, humans included, to survive changing environments across millions of years. This dual nature, occasionally harmful but fundamentally necessary, is part of what makes de novo mutations such a rich subject for ongoing research, and scientists are nowhere near finished exploring the full picture. Newer sequencing technologies capable of reading much longer, more complex stretches of DNA at once are now revealing entire categories of these new mutations that older sequencing methods could not detect at all, particularly changes involving repetitive genetic sequences and structural rearrangements, rather than simple single-letter swaps. Some of the most recent research using these advanced techniques has actually roughly doubled previous estimates of how many total genetic changes appear in each new generation once every type of mutation is properly counted, suggesting that earlier studies, thorough as they were still only seeing part of the full picture. If you find this kind of deep dive into the hidden machinery of human biology genuinely interesting, go ahead and hit that subscribe button, because there is a lot more of this kind of investigation still to come. What makes this whole area of science so compelling at the end of the day is the reminder that human reproduction, for all its apparent simplicity, is actually an extraordinarily precise biochemical process operating at a scale most people never stop to think about, copying 3 billion genetic letters accurately enough that the vast majority of the process works perfectly almost every single time, while still leaving just enough room for small, unpredictable changes to slip through and quietly reshape the genetic future of an entire family line. Every person alive today carries the accumulated residue of millions of years of these small, unexplained changes stacked generation after generation, each one a tiny accident of biology that either faded into genetic irrelevance or, in rare cases, changed the entire trajectory of a bloodline, a species, or in the most extreme cases, the course of evolutionary history itself. The specific claim of exactly 348 mysterious variants that nobody can explain does not hold up against the actual published science, but the real underlying phenomenon, the fact that every child on Earth is born carrying genetic changes found in neither parent, is true, well documented, and in many ways more interesting than the viral version of the story ever gave it credit for being. Modern genetics has spent the better part of 2 decades chipping away at exactly how this process works, moving from a vague awareness that mutations happen somewhere between generations to an increasingly precise family-by-family map of exactly how many new mutations appear, where they tend to occur, which parent they usually come from, and what role paternal age plays in the entire process. It is one of the clearest examples in modern science of how a mystery can be broken down piece by piece, not through a single dramatic breakthrough, but through years of patient, methodical sequencing work across thousands of families, until what once looked like an unexplainable anomaly turned into one of the best understood and most consistently reproducible patterns in all of human genetics. Every child born today adds one more data point to that ever-growing picture, carrying forward a small, invisible signature of new genetic change that belongs entirely to them, inherited from neither their mother nor their father, and quietly reminding us that even something as fundamental as human reproduction still has fresh secrets left to reveal. Before the arrival of affordable whole genome sequencing, scientists actually had to rely on a much cruder and far more indirect method to even guess at how often these unexplained mutations occurred. Early researchers studied rare, well-documented genetic disorders caused by a single faulty gene, then calculated backward from how often that specific disorder appeared in the general population to estimate a rough overall mutation rate for the entire human genome. This approach, first attempted in the 1930s using data on hemophilia and refined again decades later using other single-gene disorders, produced estimates that were remarkably close to what direct genome sequencing would eventually confirm, which is a small miracle considering how limited the available tools were at the time. It was not until the 2000s, when sequencing an entire human genome finally became fast and cheap enough to do at scale, that scientists could finally compare complete genomes side by side and directly count these new mutations rather than inferring their existence indirectly through population statistics on rare diseases. The actual laboratory process behind identifying a de novo mutation is more involved than simply glancing at 3 genomes side by side, and understanding it helps explain why it took so long for this field to mature. Researchers first sequenced the complete genome of the child, then separately sequenced the complete genomes of both parents, and finally run detailed comparisons, scanning every single position across all 3 billion base pairs to flag any spot where the child's genetic code does not match a plausible combination of what could have come from either parent. Because sequencing technology itself is not perfect and occasionally introduces its own small errors, Researchers have to apply careful statistical filtering to rule out mistakes created by the sequencing machine itself rather than genuine mutations, which is part of why early studies sometimes produced inconsistent mutation count estimates until sequencing accuracy and analysis methods improved significantly over the following decade. Confirming that a flagged variant is a real, genuine de novo mutation rather than a technical artifact typically requires additional targeted verification, sometimes involving specialized sequencing techniques focused specifically on the region in question, to make absolutely certain the mutation is real before it gets counted in any official estimate. One of the more striking discoveries to come out of this verification process involves something researchers call germline mosaicism, a phenomenon where a mutation does not actually appear for the first time in the child at all, but instead already existed in a small fraction of the father's sperm cells before conception ever took place. Because a man's body produces sperm continuously through repeated rounds of cell division, occasional mutations can arise early in that process and then get copied forward into a whole cluster of related sperm cells, meaning a single mutation event inside the father's body can end up appearing in multiple children if he conceives more than one child using sperm from that same mutated lineage. This matters enormously for genetic counseling, because a mutation initially assumed to be a completely random, one-time event in a single child might actually carry a higher than expected chance of appearing again in future siblings. A possibility standard genetic testing of the parents alone would never reveal, since the mutation exists only in a subset of the father's reproductive cells, rather than in his blood or saliva, which is what most genetic tests actually sample. Researchers have also discovered that these new mutations are not distributed randomly across the genome, and this uneven distribution has become an active area of study in its own right. Certain stretches of DNA, particularly regions containing short repeated sequences that the copying machinery has a harder time replicating accurately, experience dramatically higher mutation rates than the rest of the genome. Scientists have identified specific recurring locations, sometimes called mutational hotspots, where new variants appear over and over again across unrelated families, suggesting something about the underlying chemical structure of those sequences makes them inherently more prone to copying errors, regardless of which family is involved. Mapping these hotspots in detail has become an important tool for predicting which regions of the genome are statistically more likely to be the source of a newly arising genetic disorder, helping researchers narrow their search when identifying the specific mutation responsible for an unexplained condition in a child with no family history of it. The practical relevance of all this research extends well beyond academic curiosity, and genetic counselors today routinely factor de novo mutation science into conversations with families navigating difficult diagnoses. When a child is diagnosed with a serious developmental condition and genetic testing reveals a de novo mutation as the likely cause, that information can bring a strange kind of relief to parents who might otherwise blame themselves or search endlessly through family medical history looking for an inherited explanation that simply does not exist. Knowing that a mutation arose spontaneously through the ordinary imperfections of biological copying rather than through anything either parent did or failed to do has become an important part of modern genetic counseling, reframing what once felt like an unanswerable mystery into a well-understood, if still somewhat unpredictable, feature of how human reproduction has always worked. It also helps future family planning decisions, since parents can be told with reasonable confidence whether a mutation was a one-time random event or whether it carries any elevated risk of recurring in future pregnancies based on findings like germline mosaicism. Looking ahead, the pace of discovery in this field shows no signs of slowing down, and several major research groups around the world are now sequencing increasingly large numbers of multi-generation families, specifically to build more precise maps of exactly how, where, and why these mutations occur. As sequencing costs continue to fall and the technology needed to read even the most repetitive regions of the genome keeps improving, researchers expect to uncover still more categories of de novo genetic change, invisible to current methods, refining mutation rate estimates that have already been revised upward more than once in just the past few years. What started as a strange statistical anomaly buried in early sequencing data has grown into an entire subfield of modern genetics, touching evolutionary biology, psychiatric medicine, reproductive health, and genetic counseling all at once. And it stands as one of the clearest reminders that even the most fundamental biological processes can still hold entirely new layers of complexity waiting to be uncovered.